Bittersweet events for the thesis supervisor who is watching a student getting kicked out of the nest off onto a bigger and better career, but at the same time losing valuable institutional memory, intellectual skills. So it's a mixed bag of emotions. If there's 2 or 3 words I use to describe our speaker today, Brandon Russell, I use phrases like leader and eclectic and diverse in his interests. And I think the talk today will show us that facet of Brandon's intellect. But I also think if we kind of look back a little bit on his history, you're going to see this sort of meandering pathway that brings him here and probably onto some very interesting career opportunities. All right, so he grew up in Mississippi and attended starting with that. And was a Barksdale Honors Scholar at the University of Mississippi as sort of the first evidence of his intellectual skills. He got both a Carrier Scholarship and a Goldwater Scholarship, 2 very highly competitive scholarships at Mississippi. He came to MIT as one of 10 of 150 applicants for the BE REU program. I think that was the summer of 2007. 2006, if I remember correctly. And was working in my lab on a nitric oxide reactor with Vasilios Dendrolakis. And that's when we were introduced to Brandon. He applied to the DE graduate program and was admitted with an NSF fellowship, another example of his intellectual skills. When that ended, he was a fellow on the NIEHS training grant. Now, if you look at his efforts during his time at MIT, I guess the first one that sort of stood out like a sore thumb was he's an ambulance driver. He's a trained EMT, was actually head of the ambulance corps for a year or so, a rather major responsibility for a graduate student. But along the way, he also won the MIT Sloan Sales Club Bold Sell competition. Okay, and this is something we've been emphasizing in my lab. I know we've been doing it in the SMART program in Singapore, this idea of being able to sell yourself as a scientist in any light. And Brandon, I think you'll hear his talk today, has great skill in speaking, marketing, selling himself as a scientist. And I think you'll appreciate that today. But I think that award typifies a lot of Brandon. But in a more serious vein, though, he did was elected as a Siebel Scholar, which again is really for leaders. There's only 5 Siebel Scholars each year at MIT and 85 nationally. So this is a really major honor reflecting a diversity of interests, leadership skills, and significant interest in really public issues, big public issues. All right, on and on and on he goes. He participated in something called Connect to Clearview, which is a program for leadership development in consulting. And I believe you had 3 consulting offers here in Boston before you got your PhD. So once again, leadership, intellect, great skills in this regard. And I think today I'll stop and let him wow you with his very fine work in bacterial pathogens, a new area for our lab. Brandon?
Thank you, Pete. That means a lot. And thank you guys for coming. It's a bigger crowd than I expected, which means a lot to me. Let's not sugarcoat it. Bacteria are killing us. Almost. Okay. If you look globally, bacterial diseases and infections constitute about 25% of the total global disease burden. So they're responsible for about a quarter, both of lives lost and also of what are called disability-adjusted life years, a sort of quantitative metric for how much they impact quality of life in addition. not just worldwide, but also here in the United States. In particular, antibiotic resistance is a really big deal. So about a year ago, the CDC came out with a pretty fire and brimstone report on antibiotic resistance and its spread in all parts of the country, but in particular some hotspots in the southeast where I come from. And then the WHO just about a week ago put this forward, pointing out that of all the major facets of modern medicine, antibacterials and our ways to combat infectious diseases are really pretty primitive. So we have this thing called the discovery void, where we haven't had a new kind of antibiotic in about 35 years. So in terms of the pace of medical innovation, we're pretty much still using leeches when it comes to infectious diseases. So big needs here, and really big needs for science to address these things. One caveat, you probably only care about bacteria if you care about a certain subset of diseases. That's entirely fair. Infectious diseases, also autism and other sort of behavioral disorders. Cancer would be another one. If you care about metabolic disorders and autoimmune disorders, those matter. And if you care about cardiovascular disease, bacteria matter. So chances are, if you don't care about bacteria, you're probably a Martian biologist. That doesn't work either. So bacteria are important. So why do we care about that? Why am I going on and on about that? Mostly because I'm still kind of angry that my teachers lied to me. So these are DNA and RNA, and if you were like me in high school, you learned they're made up of A, C, T, G, U. These 5 letters somehow make up all the genetic code. They make proteins, do all these amazing things. Well, that's a lie. They're actually made up of these letters, about 125 of them. These are incredibly diverse in their structure, in their function, in their distribution across organisms, and for the most part, we don't know what most of these do. We find them in sort of a piecemeal, haphazard fashion. We assign functions on a one-on-one basis, one-to-one correlation. But we don't have a good systemic overview of what these things do and why they're important and why there are so many of them. So that's really the question. So you put those two things together. We have bacteria. They're really important. We don't understand them very well. We desperately need new tools for them. We have this whole set of things, this rich data set that we really don't understand well. And so you have the question that my research has tried to get at for the past several years, which is, what are these modified nucleosides doing in bacteria, and can we use them for anything useful? Can we exploit them for the things we want to do? So today I'm going to go through sort of 3 vignettes that are going to go through the stages of infectious disease. So they're going to progress through first pathogenesis, which is establishing the infection. Our goal there is going to be to see if we can find ways to disrupt the pathogenesis and prevent the infection before it even gets hold. Then you have diagnosis, actually trying to confirm what the bacteria is, figure out the best course of treatment. The idea there being if you can enhance, find a more rapid, more sensitive diagnostic, you can get therapy before some of the deleterious effects have set in. And then finally, we'll conclude by talking about therapy. The idea being, can any of these modified nucleosides be targeted as ways to adjuvant or enhance antibiotic therapy? So in each case, we'll talk about a different type of modified nucleic acid, and in each one we'll talk about a different type of bacteria. In each case, the particular things chosen to really highlight the key features, either highlight pathogenesis, highlight diagnosis, or highlight therapy. So that's the high-level overview. We'll dive right in. So first thing I looked at was the role that these modifications, specifically these modified nucleosides in RNA, are playing in a pathogen called Helicobacter pylori. And this started sort of from the global health perspective. The Helicobacter pylori is the world's most pervasive and most successful pathogen. Infects about 50% of the world, so about 3 billion people globally are infected with H. pylori. The vast majority of those are not in sort of the Western developed world. As you can see, the United States and Europe are comparably light-colored, but instead they disproportionately affect the underdeveloped nations. In particular, Southeast Asia, South America, Sub-Saharan Africa are really disproportionately affected by this organism. Persists for the life of the host. It's incredibly well adapted to its niche, which is actually in the human gut, which for a long time was thought to be sterile because you have gastric acid and you have the constant motion and digestion of food. So it's sort of difficult to imagine something living there, but H. pylori manages to do so quite well, and well enough that it's actually undiagnosed and asymptomatic in the vast majority of cases. So about 90% of people infected with it don't know they're infected. But the problem there is even when you have that low-level infection without overt symptoms, it's actually a group 1 carcinogen. So it significantly predisposes you to gastric cancer, which if I didn't know this before I started studying, it is actually still the 4th leading cause of cancer death worldwide. So it's a really big problem. And again, in particular, Sub-Saharan Africa, South America, Southeast Asia all have these huge levels of gastric cancer, a lot of which you can track back to H. pylori. The other problem, in addition to that great adaptation, is that diagnosis is relatively simple, but treatment can be complicated. Most treatments involve 3, 4, 5 therapies concurrently in order to knock it down. And there's a pretty deleterious effect where the process of treatment actually synergizes, and you get something called phenotypic resistance. So basically, as you start to knock down the population, you end up with fewer bugs. The pH of your stomach starts to drop because they're no longer buffering it with their sort of active evasion. And then that acidic stress can actually induce drug resistance in a small persistent population of the H. pylori. So we really need ways not only to target them better, but to find ways to disrupt that antibiotic resistance and that phenotypic resistance that it experiences by being able to evade the host immune system. And it's exquisitely well adapted to doing that. So this is basically a cartoon showing all the things that H. pylori has to pass through and get through and survive in order to productively infect the stomach. So the first one being that it's a pH of about 2, it's very acidic. And so it has a number of enzymes expressed on its surface that produce things like catalase and urease. Basically makes a little envelope that buffers the local pH and allows it to burrow into the mucus and get under the mucus close to the cell lining where the pH is more favorable and where it's able to depolarize cells, pull nutrients out, evade the innate immune response, all that sort of thing. So it's an incredibly complex, highly orchestrated process that allows it to do this sort of evasion. And yet despite this, compared to other bacteria, and in particular ones of similar genomic size, H. pylori has relatively few transcription factors and well-known transcriptional regulators. So the sort of traditional way we would think of controlling these sorts of large-scale things, most of them seem to be missing in H. pylori. So this sort of piqued our curiosity and said, well, if it's not using transcriptional regulation, how's it doing things? And this led us to a model that was developed by some researchers in our lab and some collaborators at the University of Albany, where RNA modifications, which had traditionally been thought of as sort of static, structurally integrity-holding modifications, can actually participate in dynamic stress responses. So it's a bit complicated, but the way this basically works is you have a pool of tRNAs, and you have a 3-nucleotide anticodon loop, which is actually what binds to the mRNA. And traditionally, the first 2 nucleotides are the ones that bind tightly to the mRNA. The third is called the wobble position. It doesn't bond as tightly, and so it enables you to have a little bit of flexibility with which tRNAs pair with which mRNAs. It's possible to modify that wobble position by doing some sort of nucleic acid modification, a methylation or a hypermodification where you add on a lot of different atoms and change the structure pretty significantly. And that wobble-modified tRNA actually binds more tightly to a specific cognate mRNA, so one particular codon that tRNA codes for. And then what you do is if you have mRNA transcripts that have a statistical overrepresentation of that particular codon, say you had 10 lysines and say you had 3 codons that could code for lysine, you'd expect 3 of each if you were just randomly distributed. But you might have a case where all 10 of them are coded for by this one particular codon that could be biased by this modification. So what you end up doing is you have a translational thermodynamic advantage where these mRNA transcripts get pulled to the front of the line and preferentially translated because you have better binding, better kinetic efficiency with the tRNAs. So this has now been demonstrated in a couple of different organisms, well characterized to occur in yeast, well characterized to occur in human cells. Some of these modifications appear to be tumor suppressors, so they're clearly doing really important work, but no one's ever shown one of these in a bacterial pathogen, which was interesting to us, especially considering the fact that clearly H. pylori is doing something past the transcriptional level. And so we thought it made it a very good candidate for this sort of translational regulation by these RNA modifications. And so that was the goal. The basic pathway here is you take your organism of interest, in this case H. pylori, you stress it with stress of interest, in this case the things that it's evading in the innate immune system. So things like hydrochloric acid stress or hydrogen peroxide produced by neutrophils in the innate immune response. You isolate the RNA, process it, and then you use high-resolution mass spectrometry to characterize those modifications. So you have a full set of these, somewhere between 20 and 30 occur in any given organism. You define what those are, and then you go back, you do defined endpoints, 20% lethal stress, a 50% lethal stress, and then you quantify those nucleosides using high-accuracy mass spectrometry, so triple quad mass spec. And what that lets you do is you can calculate fold changes in those exposed conditions relative to the unexposed conditions. And that can give you clues as to which of these modifications might be participating in stress response and be important in survival. So that's the idea. The first thing to do was basically to standardize what stresses we were going to look at. So hydrochloric acid made the most sense. That's the first thing H. pylori has to survive in order to invade the stomach. After that, the most important reactive oxygen species is probably hydrogen peroxide, because that's produced in pretty huge amounts by neutrophils at the site of the local inflammation. And H. pylori is exquisitely well adapted to evading both of these. And so we know that there are specific enzymes that must be upregulated to survive these stresses. And so we did some dose response curves so that we're looking at common phenotypic endpoints. The only interesting thing here is that you see a hormetic effect, where at low dose for both of these stresses, you actually increase the survival of the organisms, likely because you're sort of tickling them and you're setting off a cascade of stress response and cell survival proteins. So that's consistent with what we know about the niche and how they grow. So following exposure, we did some chromatography. This was in collaboration with a former postdoc in our lab, Kok-Chong Lim, and a former UROP student we had, Sebastian Smick. And so together, using a couple of different types of mass spec, we were able to generate a candidate beginning list of the modified nucleosides in H. pylori. You'll notice 17 is less than 20, it's much less than 30. So we know these are not all of the modifications. And in particular, there are a couple that we know occur in bacteria, we know H. pylori has the genes for, we simply don't see them. They're likely at a level sufficiently rare, only occurring on one tRNA, that they're below our limit of detection. But it's a sufficiently large spectrum and it includes enough sort of unique modifications that it's very much amenable to this sort of profiling. And we can see how these things change in stress and whether those changes correlate or give us any sort of clues as to the mechanism. And so we did that, and the answer is yes, actually. So what we have here is a heat map. These are normalized fold changes of the exposed cells relative to the control cells. And they're plotted as a heat map and clustered so that the two hydrochloric acid stresses cluster together, the low dose and the high dose, and the two hydrogen peroxide stresses, again low and high, cluster together. And then if you look, you can see that you get a bifurcation right about here where things that are highly upregulated in the acid stress are downregulated in the oxidative stress, and vice versa. And so this was encouraging, this makes sense, because these two have very distinct mechanisms of toxicity, and they have distinct enzymes that are necessary to detoxify and survive.
Pull that drape shut a little bit.
Yes, I can.
Is that better? To see the slides.
Yeah.
Is that better?
Probably.
Yeah? All the way?
There we go. All right. Thank you, Steve. So the answer is yes, these things do appear to rearrange and reprogram in specific ways that are unique to the stress conditions. So then the next question was, are any particular modifications important? And so to do that, we use principal component analysis, which is a multivariate way of pulling out hidden patterns in data without getting too into eigenvalues and eigenvectors. Essentially what it does is it groups things based on similarities in the underlying data set, and it tries to find synthetic variables that account for the variance. In this case, two principal components account for almost 90% of the variance, which indicates that you can explain a high degree of these differential responses with a relatively small number of combinations. And then if you compare the left-hand plot to the right-hand plot, essentially things in similar locations on the left are important for things in similar locations on the right. And so we see the group of hydrochloric acid is very tightly grouped in the bottom left corner. Things like M62A, M3C, M1A seem to be associated with that sort of stress. And then, interestingly, principal component 1 is almost purely to separate the low-dose hydrogen peroxide from the rest of the stresses, which likely is a result of the fact that H. pylori is well adapted to its niche and is sort of used to surviving a little bit of hydrogen peroxide stress. And then principal component 2 well separates the high-dose H2O2 from the low-dose HCl. So we have really good indicators here that there are some particular modifications associated with surviving these particular stresses, which is really cool. And it's actually the first time that's been reported in any sort of human pathogen. So prior to this, this was only reported to occur in yeast and humans. So to quick recap, this is the first time anyone's done a profiling like this of all the modifications in H. pylori. So despite not being a full set, it's the first time someone has put together a catalog of them. It's the first time we've demonstrated this sort of translational control, even the possibility of it occurring. So it's the first time we've seen this dynamic reprogramming of modifications in a pathogen, and it's the first time particular modifications have been associated with particular stresses. And in particular, some of the ones that come out have interesting functions and have some potential that they might actually be mechanistically linked. So M62A, for example, for a very long time was thought only to occur in ribosomal RNA. It was thought of as a contaminant in tRNA. And then a few years ago, our lab actually proved it occurs in the tRNA of mycobacterial species, which are also renowned for their ability to survive and persist in really harsh, inflammatory, oxidative environments. And so that sort of piqued our curiosity that this might be playing a role in sort of oxidative stress in general. M7G has consistently been shown in all organisms to be necessary for robust growth, particularly under temperature stress. And then MS2T6A is interesting because it's only known to occur at position 37. So this is one adjacent to the wobble position. And that's a place where many modifications have been shown to modulate the way that this enters the ribosome, binds to the mRNA, and then leaves the ribosome. And so these are all interesting, and they point to the fact that these might actually be doing important things at the level of response. And so moving forward with these, a couple of important things to do. You can generate knockouts of the associated enzymes. So the particular strain of H. pylori we use is fully sequenced, and so we know which genes are responsible through homology searching for most of these modifications. You can do targeted knockouts, and you can retest and see if you induce sensitivity. And then if you do, then these might actually be important targets for potential antibiotics, adjuvants, or even ways to prevent the infection without decimating the rest of your gut flora, which is an important thing, because there is a little bit of evidence that sort of dispassionate elimination of H. pylori is not always a good thing for health. So this was sort of really encouraging. To sum it all up, we have really good evidence that these RNA modifications are important in pathogenesis of bacterial infection and might be useful as modulated targets. So, we next wanted to sort of step things up. We established that these are important in establishing infection. Are they important for us, clinically, in figuring out what the infection is, tracking the infection, that sort of thing? And so for the next set of studies, we came at it from the other side. So for the first ones, we were really started out motivated by the global health implications, interested in that particular bacterium. For these questions, we were motivated from the other side, which was we thought RNA modifications had potential as good urinary biomarkers. There were a couple of reasons for this. The first one, this graph on the right, is that these modifications tend to group in patterns. So you have some that are what we call universally conserved. You find these in eukarya, bacteria, archaea. But then you have large numbers of them that are unique to eukarya, unique to bacteria, unique to archaea. And even then, within certain kingdoms, we have evidence from some of our collaborators and some of our own work that some of these are unique to a particular genus or even a particular species, possibly. And so you have something that might be unique to, for example, a human pathogen. The other thing that's interesting is that it's well known that there are salvage pathways in virtually all organisms to save nucleosides. So you have cell turnover, you can reuse RA, RG, the sort of normal nucleosides so that you don't have to de novo synthesize everything and waste a lot of cellular energy that way. But we know that there don't exist salvage pathways for most of the modified nucleosides. And so for a very long time, people have looked at these as cancer biomarkers in the urine. The idea being that when you have rapid cell growth of cancer cells, you have huge numbers of these modified nucleosides getting excreted in the urine, and you can sometimes correlate patterns. What we were coming at it from was a little bit different, was that if you have a bacterial infection, bacteria are turning over, they're naturally being killed, and the DNA and RNA are going to come out, and they have to go somewhere. And if you have modified RNAs that aren't going to get recycled by these salvage pathways, they're probably going to come out in the urine of the infected organism. You got the big sugar moiety, makes it water soluble. So the question was, can these serve as urinary biomarkers? So we looked and we said, well, we need an organism to look at. We need an infection that we're interested in trying this out on. And so we said, what organism or what infection is important, has biohealth implications, but has problems with diagnosis. And so we pretty quickly came to the world's second most common infection, tuberculosis, which, like H. pylori, is unequally distributed across the globe and is a significant global health impact. This one is hard to argue with the numbers. So about 10 million new cases of TB are diagnosed every year. About 2 million people still die from TB. And every year that goes by, the number of TB-resistant mutants actually goes up. So we're at a point now where about 5% of all those new cases of TB are what are called MDR, multi-drug-resistant TB, that don't respond to the 2 first-line therapies. XDR TB, extensively drug-resistant, is about 0.8% of new diagnoses. And then there are scattered reports from a couple of different continents of TDR TB, totally drug-resistant, where you have absolutely no known antibiotic that works. So in addition to being very difficult to clear, it's also really difficult to diagnose, particularly at a level when it's still susceptible to therapy. So the gold standards, according to the WHO, are sputum smear microscopy, where you actually have the person cough, you look at it under a microscope and see if you can find bacteria, or culture of the sputum. And both of those suffer from the fact that they're not terribly rapid and they're not terribly sensitive. So sputum smear microscopy in particular, you only get a positive signal on that once you have about 10 to the 3rd bacteria in the lungs. So you have to have a pretty significant bacterial burden before that shows anything. And then culturing can take a couple of weeks, can even take up to a month to get the answers back from that. So there's not a good, rapid, sensitive diagnostic out there. So we looked at that and said, all right, do RNA modifications function as a good diagnostic? And then we ran into one problem. Tuberculosis is a BL3 organism, and we do not have a BL3 lab here. Turns out we're not the only ones that suffer from that problem. So BL3 labs are comparably expensive and they're comparably rare. In contrast, virtually any laboratory with benches and hoods can operate at the BL2 level. And so because of this, BSL-2 models of tuberculosis are actually pretty common in tuberculosis research and in the literature. The most common one that's referenced is this zebrafish model. So basically, zebrafish get a version of tuberculosis caused by Mycobacterium marinum. It's basically fish tuberculosis, and it recapitulates some of the features of human disease and is used as an easy-to-use, easy-to-handle BSL-2 model. The only problem being that the utility of this, particularly for things like diagnostics or drug development, is really limited by the biology. So fish don't have lungs, which is one of the biggest things, tuberculosis being a lung disease. It's also really hard to get urine or feces or things like that that are clean from a fish model. So it's not really amenable to biomarker work, not really amenable to diagnostic testing, and certainly not amenable to the sort of pharmacokinetics and drug development work you would want to do. And so we looked and we looked and we looked, and it turns out there's not a BL2 model of TB in an organism that's sort of pharmacologically tractable, that might be useful for things like biomarker screening or drug development. And that turns out to actually be really important. So TB, unlike a lot of bacteria, is really, really hard to get rid of. So antibiotic therapy for TB is standard 6 months, can last for a year, can last for 2 years in the case of these extensively drug-resistant strains. So that's 2 years taking an antibiotic every single day to get rid of the bacteria. And the main reason for that is it's really tough to actually get the drug to the bacteria because of the biology of how TB works. So essentially, you take a drug, it goes through the bloodstream, you have standard pharmacokinetics. It has to get from the general circulation into the lungs, and then once it's in the lungs, things just get more difficult. The defining feature of tuberculosis in the human is the formation of this granuloma, which is a walled-off cellular structure. It's the body's way of trying to keep the infection at bay, and it's surrounded by a tough fibrous cuff, and it tends to be poorly vascularized, and it tends to be poorly perfused, which means you're going to get relatively low penetration of the drug into the granuloma itself. And then even once you get into the granuloma, getting into the bugs is difficult. Mycobacteria are renowned for their very thick, waxy cell wall that's tough to penetrate. And even then, a significant fraction of your mycobacterial population is going to be dormant. So these are non-replicative, but still viable cells that have low metabolic activity. And so once all is said and done, you finally get your drug in, you have your molecular target, and it can take effect. And so the thing is, this takes a very long time to do. And what it means is that the sort of standard pharmacokinetic parameters that we use for evaluating new drug entities don't correlate well at all with efficacy against TB. So if you look at tables of pharmacokinetic parameters for effective TB drugs, the values are all over the map. They don't follow the sort of general rules. And so there's increasingly this recognition that TB drug development, even compound lead optimization at a relatively high level, needs to be done in some sort of animal model. You need to have some proof that what you're trying to do is actually going to make it to the granuloma. But BL3 facilities, being costly, being cost prohibitive, tend not to be amenable to these sort of large-scale screening studies. So not only did we need a BL2 model to do our own diagnostic and biomarker work, but it seems like there's a great need for a BL2 model in general that's amenable to these sort of large-scale screening studies. And so that was what we set out to do, was to create a BSL2 model of TB in an animal that's pharmacologically tractable. So we chose Mycobacterium bovis, BCG. This is the vaccine strain of TB that the vast majority of the world, other than the United States, uses to vaccinate people against tuberculosis. It's an attenuated version of basically cow tuberculosis created by serial passage in vitro, so it's lost some of its virulence determinants. But it's still about 99.9% sequence identical to Mycobacterium tuberculosis, and in particular, a lot of the sort of characteristic antigens and characteristic proteins of the Mycobacterium complex are still present in BCG. So it made it a good model. We use endotracheal infection that was developed originally by scientists at Novartis in their tropical disease lab, in particular the lab of Pablo Bufani. This is different from a lot of studies which do things like aerosol, or you put the animals in a cage and then you pump in TB and they breathe it in. Those tend to recapitulate the actual process of infection better. They're good for transmission studies, but you get all over the map levels of the infection. They're not reproducible, and you don't have good control over the amount of the infection. In contrast, the method that we use, you can develop a known quantity of the bacterium directly into the lungs, and you can get highly reproducible, highly controllable infection. And then the final thing that we did that's important is we used an animal known to be pharmacologically useful, the rat, it's the workhorse for pharmacokinetics, and we used outbred strains. So a lot of TB models that you'll find in the literature use inbred strains, which are really nice, they're controlled, they have predictable response, but they don't well recapitulate the fact that TB in humans has pleiotropic outcomes based on host factors. Whereas we use outbred animals, so you get a little bit of genetic variability within the animals themselves, and you can recapitulate some of that unpredictability in the outcome. So the study timeline we use is at day 0. We have 34 outbred female rats. we dose them with either a sham control, just PBS for our uninfecteds, or we dose them with a high dose of BCG. Being an attenuated strain, this is consistent with the literature. Usually a high dose is used so that you can actually get that infection to take hold. If this were real virulent TB, it would be about 4 orders of magnitude lower. And then we sacrifice 4 of them to actually ensure that the infection took hold. So then we're left with our cohort of 30. And we do weekly necropsies and collections of urine for the biomarker stuff, 2 controls, 4 infecteds, and then we track the various infection parameters over time. In particular, we're looking at the burden of infection, the degree of immune infiltration into the lung cells, the plasma and tissue cytokine response, and then the amount of pathology, whether these develop the sort of characteristic TB lesions and TB lung pathology that you would expect to see. So, going through, the answer is yes, these things do establish an infection quite rapidly and quite sufficiently. So here on the right, this is the day 1 cohort. So basically, if you sacrifice the animals 1 day after establishing the infection, you can recover virtually the entire inoculum in the lungs. So we know that the full dose is actually getting into the lungs. And half of the animals actually have dissemination to the spleen at day 1, which is a marker that the immune system is working well, that these animals are disseminating, they're perfusing, and it gave us hope that this would actually be valuable as a way of checking the immune response. And then if you track the infection over time, you see that it generally goes down. We do get a little bit of a bump between weeks 2 and 3. This is consistent with the fact that we start with that high dose inoculum, so there's sort of not a lot of room for it to grow, but it does sustain quite well, very well above the limit of detection, all the way out to week 8. And you get relatively little dissemination over the course of the weeks into the spleen. So all totaled, only about 6 of the 24 dosed animals ever had any recoverable bacteria in the spleen, which indicates it's a relatively contained infection within the lungs. So spleen dissemination is a good marker for severe disseminated disease. And since we don't see a lot of that, it indicates the disease is primarily contained within the lungs, which is consistent with them being able to control, wall off the infection like you see in the majority of human TB cases, which was encouraging. If you look at how the lungs respond, you see that at weeks 3 and 4, a number of immune cells infiltrate the lungs at a really high degree. So we looked at 4 particular types of immune cells. Definitions are on the right for those of you unfamiliar, like I was when I came into this. So basically, granulocytes are a form of the innate immune response, and then the other 3 are either adaptive immune cells or they're linkers between the innate and adaptive immune response. And what we see is that you don't get good infiltration and good invasion until weeks 3 or 4, which is consistent with the tuberculosis literature, that it actually takes a pretty good amount of time for the bacteria to get the lungs, replicate to a high enough level that they're taken up by the resident immune cells, trafficked to the local lymph nodes, you drive differentiation of T cells, and then finally, after about 3 or 4 weeks, these migrate back to the side of the lungs, and that's exactly what we see, that at weeks 3 or 4, we have significant upregulation of the immune cells that you would expect to see. In particular, we see the CD3 positive T cells that are known to be upregulated late in the TB infection, and we see these co-positive cells down here, which represent tissue-derived antigen-presenting cells. So we know that the cells present there in the lungs are the ones taking these bacteria up and driving this differentiation. So all consistent with what we see in human TB. And then the next thing we looked at was a cytokine panel. So this is a multiplexed assay for 23 different cytokines that are present, both pro-inflammatory and anti-inflammatory. And we looked at them both in the serum, so in whole blood that we took, and in the lung tissue itself. Okay? And what we did was we took the last 2 infection time points based on the fact that we only see the heavy infiltration at the late time points. So we took weeks 4 and weeks 8, and once again we did PCA, like I talked about earlier. And what we do this time is we pull out the infected group, it's these blue circles on the top right, and the uninfected group, these green circles on the bottom left. And once again, if you look at the factors that are associated with each group, you see things that make a lot of sense and that are consistent with human disease. In particular, the infected group seems to be heavily associated with high levels of macrophage colony stimulating factor, which is consistent with the fact that macrophage is the preferred niche for the bacteria to replicate, as well as some pro-inflammatory cytokines like IL-2 and IL-17, which a lot of literature point to being very important in driving the pathogenesis and making for the sustained infection. And then in contrast, you see a couple of pro-inflammatory cytokines being reduced in the uninfected animals. In particular, IL-1 alpha and interferon gamma are very important in TB pathogenesis. So the fact that we see those being predictive and pulling out the uninfecteds makes a lot of sense. So the immune infiltration matches, the infection is at a high level, and the cytokine response matches. What does the disease actually look like? And this is when we got really excited. So on the left here, this is a control example of just a healthy rat lung. And then down here, this is 2 weeks rat infection with actual virulent TB. This This is a virulent clinical strain of TB. And the important things here are you see the beginning of granulomas, which are these dense pockets of immune infiltrates, dense cellular structure with a well-defined wall. And you also see a lot of what's called just sort of general cellularity. So you should have this very open structure, but instead you get this dense invasion of a lot of immune cells and also a lot of bacteria. And we see pretty much the same thing in our animals. These are 3 successive micrographs from the same animal. This is 1 week post-infection. And in each case, the blocked off area is zoomed in the subsequent slide, or subsequent micrograph. And I've highlighted some of the important features with these numbers. In particular, we see tons of inflammation, and we see tons of cellular invasion, and we see the beginning of well-organized foci of granulomas. We also see some foci of mineralization, which indicates that we're beginning to lose perfusion, and we're beginning to get that sort of necrotic or caseous or sort of not well vascularized core, which is really nice to see. And then the other really interesting thing is here, this is the bronchiolar wall, and this should be a single sort of uninterrupted line of cells, but instead you actually have attenuation of the wall by this granuloma, and you have sloughing off into the airspace of the immune cells and presumably also the bacteria, which is actually how the disease is transmitted. So if this animal were to cough or to sneeze, it would pull this infiltrate, this invasive material into the airspace, cough it out, sneeze it up, and actually be an infected animal. And so all of these were really impressive, especially given the fact that from a gross pathological level, the animals don't seem terribly sick. So it's not a severe disease, their body condition score doesn't change. It seems to be relatively well contained into the lungs, but it recapitulates most of the pathology that you would expect to see in human TB. And so we got a pathologist to sort of grade these. So each case you have the weeks, and each animal is represented by a single bar, and we have just a qualitative grading scale of the degree of pathology. And so what you see is even out to week 8, when you have the low-level bacterial load, you still have a really high degree of pathology in all the infected animals. And that was really nice to see because it indicates that it's actually a severe disease, and again, that you see this sort of large-scale reorganization and restructuring of the lung, a lot like you see in human TB. So at this point, we had a nice model, and we had a lot of the features of human TB. In particular, some things I haven't mentioned before, we see a lot of multinucleated giant cells, which are a particular marker of TB in the lungs. And we also see those mineralization foci that indicate that we're beginning to get this loss of vascularization, this loss of permeability of the granulomas, which is nice because that indicates they might work as these sort of models for drug targeting. So we finally had our model, and then the question was, can we do this urine biomarker idea? So without going too much into this, a lot of urine studies are very bad because they are not well controlled. They do things like room temperature collection of urine, they do 24-hour collection, they do collection with the food and the feces, and then they report gobs and gobs of damaged biomolecules in the urine. No big surprise there. So we were very careful to do a very controlled study. We fast the animals so you don't have food pellets. We add an additional filtering step so you have very little feces in the urine. They're frozen immediately because the containers are suspended in dry ice, the urine collection. So we get really clean, fast frozen urine. Then we minimally process it. We basically just centrifuge it down. We add an antioxidant cocktail that we developed in our lab for our DNA and RNA work. And then we do a quick digest because we had some evidence that full-length or longer strands of oligos can actually persist in the urine. And we wanted to look at single nucleosides in particular. And so then what you end up with is this clean urine with these digested nucleosides, and you can analyze that by liquid chromatography and mass spectrometry. You do that, and it turns out urines are quite common. Nucleosides are quite common in the urine. You see a lot of species. So in the top panel, these are species that have lost the mass of 132 Daltons. That corresponds to losing the ribose moiety. So these are putative ribonucleosides. This is an untargeted scanning experiment where we're looking for things that have the same sort of structural mass transition as a nucleoside would. And then in the bottom panel, these things lost a mass of 146, which would correspond to a 2-O-methyl modified ribose. So these are the 2 most common classes of ribonucleosides. All total, we see about 42 signals that lose these masses. And then if we go to databases of known modified nucleosides, we can actually confirm about 12 of these species based on their M/Z value and their retention time. And then we can further confirm 8 of them by comparing them to synthetic standards that we have in the lab. And so you end up with a lot of species that either don't match any known modification and might not be nucleosides or might be novel ones. And you also end up with a lot of species that match, but we don't have the synthetic standards in the lab. So this is really nice to see. There are a lot of these things. So do they correlate? The answer there is no. they don't correlate with infection. So we looked at two different things. Number one was we looked for the sort of gold standard biomarker, where you have something present in the infected cells, not there in the uninfected, and we don't see that. Nor do we see the inverse, where you have something only present in the control that maybe gets knocked down by the course of infection. So all these species can at some point be found in both the infected and the uninfected animals. The other thing we tried to do was to see if we could find patterns of changes. So here, this is the same data plotted both as a heat map and as a PCA, like we've talked about before. And you don't see any nucleosides being predictive of either infection or uninfection status. And this is pretty consistent with what's seen in the cancer literature. So cancer biomarkers in urine have been tried for a number of years, and what you end up finding is that interperson variability is far and away the driver of these patterns of excretion. And we find the same thing, that within a single animal, the pattern of excretion varies wildly compared to its sort of animal cage mates and even the matched controls. And so the answer was the urine biomarker idea didn't pan out. But the model itself worked very well, and we still have a large untapped data set that we could look at. So here, for purposes of being certain of what I was looking at, I'm only profiling the confirmed species and doing the pattern recognition of those, and those don't pan out. But it is possible that there are other species, either that we didn't target or that are previously undefined, that might correlate with the infection. So we have the first report of a pharmacologically relevant BL2 model of TB. It's really nice not only for our purposes, but could actually be useful for other people and other labs, and in particular, small startups that don't have access to the sort of cost-prohibitive BL3 facilities that are becoming increasingly needed to do these screening studies. And we also have some really cool insights into granuloma formation. So this is a little bit of nitty-gritty TB, but basically there are still debates about what factors are actually necessary for granuloma formation. And one of the ones that gets implicated a lot is this antigen from TB called ESAT6. It's an early secreted antigen. It's not present in BCG. It's one of the virulence factors that was lost in the process of pathogenesis. And for a long time, people said, well, the reason BCG doesn't form granulomas is you don't have this protein. But we get pretty good examples of granuloma formation. In contrast, mice tend not to get granulomas even when infected with virulent TB that has that protein. But mice consistently lack MMP1. So this is an extracellular matrix metalloproteinase that has been implicated in some recent studies in being necessary to sort of restructure the lung and allow granuloma formation. Our rats do produce MMP1, and we get these granuloma structures. And so we have pretty good evidence here that ESAT6 is not always absolutely necessary for granuloma formation, but these extracellular proteinases do appear to be important for restructuring the lung. And then the last thing is we proved that we can use this animal for these biomarker studies. It works well for pharmacological studies. And our initial hypothesis didn't pan out, but we still have the useful model and a couple of more data sets that we can pull into. And so in the end, we have this new model that we think will actually really help accelerate both drug and diagnostic development for TB. So we know how infection is established. We know bacteria involve RNA modifications there. We know that they have potential handles for biomarker and diagnostic applications. Final thing we wanted to do is we wanted to really pull this together and see if we could find something clinically relevant for a DNA or an RNA modification. And for this, again, we sort of came at this unexpectedly. And this is actually a DNA modification. And this story goes back about 25 years. So the first observation of this modification was a few people in the lab trying to do a standard tris electrophoresis on some extracted DNA. And they found that they got a schmutz. So basically you got degraded DNA here on the left. And through really good biochemical work and really good genetic detective work over about 20 years, it was discovered that certain species of bacteria harbor a sulfur modification in their DNA. It was eventually found that this is actually something called a phosphothioate, which is odd because that's an old synthetic modification. It's been known at least since the 70s that you can put into synthetic oligonucleotides to stabilize them against degradation or for antisense applications. And yet it turns out bacteria were doing this the whole time we thought we had invented this. So in bacteria, this is conferred by a 5-gene cluster called DND for DNA degradation. And it's put in site-specifically and stereospecifically. So you have two diastereomers, an SP and an RP. Bacteria only make the RP, and they put these in at nucleotide consensus sequences. So between two nucleotides that are part of a larger consensus sequence, which is really cool. And these things are found in tons of bacteria. More than 200 species have been identified. And they're always found on mobile genetic elements, either large-scale plasmid insertions or integrative conjugative elements. And they differ wildly between these different strains in their recognition sequence, in their frequency, and sort of in their derivation. And so it's clear that these things are getting shuffled around through multiple independent events of gene transfer rather than inherited sort of large scale from a single progenitor. And they're found in a lot of clinical isolates, which sort of piqued our curiosity. What they do is a little bit harder to figure out. So a few years after that, it was found that they function as a restriction system, so sort of a classical restriction modification system. Basically, you have in the top panel bacteria that have this modification, in the bottom panel bacteria that lack it, and then you're transfecting those bacteria either with a plasmid lacking sulfur, that's on the left, or possessing sulfur on the right. And so bacteria that have this modification are capable of keeping out DNA that lacks the sulfur modification, whereas bacteria that don't harbor it, don't care, they'll take up either piece of DNA. And it was eventually found that there is an additional set of genes, a 3-gene cluster, D and D, F through H, that actually perform this restriction. So it's found clustered along with there are 5 genes that we know about, found on the same mobile genetic elements, responsible for this restriction. Which is a nice, clean story, except for the fact that the majority of the organisms that we've looked at don't have these restriction genes. So in about 150 of the 200 known sequence D and D strains, you don't have any restriction enzymes. So you don't have F, you don't have G, you don't have H. And so clearly there's a reason that the cell is expending the biochemical energy to put in these modifications if it's not sitting around and participating in the restriction modifications. We got our first hint that it might be something useful and interesting for clinical applications a couple of years ago when a study came out and showed that in organisms lacking the PT modification, they're sensitized to hydrogen peroxide stress. Or, put another way, organisms possessing the sulfur survive better under hydrogen peroxide stress. And so the idea here was that it's actually acting, the DNA modification itself, as sort of an antioxidant shield. The hypothesized mechanism is that you have your normal phosphorothioate DNA, an oxidative hit comes in and cleaves off the sulfur, and then you're able to regenerate the standard phosphate backbone rather than inducing a strand break, which is what would normally happen if you had an oxidative hit at the normal phosphate. And this was really interesting to us because hydrogen peroxide, as we talked about a little bit earlier with H. pylori, is a key feature of the innate immune system. And so if you have bacteria that are shuffling around this DNA modification that enhances their ability to survive oxidative stress, this might be giving them an advantage in clinical isolates, might be giving them resistance against the innate immune system. And there's a little bit of data, controversial in the literature, that it might even be predisposing them to antibiotic resistance, because there are some reports that oxidative stress is necessary for antibiotic function. And so this was really interesting to us. So we came to the question of, do these modifications participate in antibiotic resistance, and do they exert their effects sort of pan-species? So this was done in a single organism using a single set of D and D genes, and we wanted to see sort of how deep the rabbit hole went. Does this actually pan out as a general transferable mechanism of resistance? So we looked at 3 different organisms, and we tried to span both artificial and native D and D, as well as those with and without the restriction system. So the first is a very well-controlled system, Citrobacter rodentium. It's basically mouse E. coli, and it lacks PT. And so we have an artificial system where we put in a plasmid containing the 5-gene cluster from Salmonella, which is the original organism sequence. Then we have Esenterica, which is the exact same organism used in the study identifying the hydrogen peroxide resistance. This one naturally has the full 8-gene cluster, so it has restriction. And then finally, we have a native system lacking restriction in the form of Streptolipidans, which is a soil bacterium that has a unique D and D cluster. So it's not the same one as in either of these two, has a different consensus sequence, different spots that it puts in the PT modifications. And in each case, for each organism, we looked at hydrogen peroxide as sort of our control, since we know there's evidence for that sensitivity. And then we also looked at a couple of different antibiotics, in each case guided by whatever the existing resistance pattern of that organism was. So in this case, both Citrobacter and Esenterica are resistant to beta-lactams, so we didn't look at those. And then Eslibidans, the inverse, is resistant to the aminoglycosides. We didn't look at those. And in each case, we did 2 types of assays to quantify the effects. One is the growth curve assay, which will visualize both growth inhibition as well as active cell killing. And then we also looked at a spotting assay, which will specifically visualize cell death. And that allows us to tease apart the mechanism to see if these things are actually just overcoming the inhibitory effects or if they're actually allowing these to survive the stresses. So So the first thing we did was we proved that adding or subtracting PT doesn't do anything under normal conditions. So a really important control that all three organisms grow equally well with and without the PT modification. And that goes for whether you're artificially inducing the PT or artificially deleting the PT. So no effect. And then for each one we step through, and we're going to look at some representative graphs, representative antibiotics. These data are highly reproducible, so we sort of picked the illustrative ones. In Citrobacter, which is an artificial set of PT genes, no restriction modifications, you induce sensitivity to hydrogen peroxide. So this is the exact opposite of what we expected to see. This is sort of our first hint that something pleiotropic and something unexplained is going on here. So in contrast to the salmonella results, you actually end up with cells that are much more sensitive to hydrogen peroxide stress when they have the phosphorothioate. And then in contrast, you end up with a growth advantage under antibiotic stress against any aminoglycoside with these PT-positive organisms. And so we did see this antibiotic resistance phenotype, but we didn't see the oxidative stress resistance that we expected to, which was really odd. So we went back and said, okay, we're going to pull the exact same strain used in the paper, make sure we can actually do it in our hands. We can. You take the same salmonella strain, expose it to hydrogen peroxide. As expected, PT confers resistance. Lacking PT, you don't have as robust of growth. And then you have absolutely no effect across the bevy. You have about 6 antibiotics we looked at all totaled. Your sensitivity to the 2 strains is absolutely identical. So things are clearly going on that we don't understand. And then to sort of tie things together and complicate the story even further, if you have another organism that naturally harbors PT and lacks modification, you end up resistant to both. So in Streptococcus, which is the soil organism with a different PT D&D set of genes, you end up resistant to hydrogen peroxide stress and resistant to antibiotic stress when you have PT. And so we knew something interesting was going on. And we knew that the effects of PT were clearly not sort of generalizable. They are organism specific, that the particular sequence and source of the D&D genes clearly matters, as well as toxicant specific. So the type of stress that you're inducing matters. And so we wanted to know, is this purely an inhibition phenomenon, or is cell death actually playing a role here? And so this is a lot like a yeast dilution plate assay. So essentially you have increasing dose of the toxicant as you go up the panel, and then you have technical duplicates of either a PT positive strain on the left or a PT negative strain on the right. And then we've got 2 panels of Citrobacter and then a panel of Salmonella. And what you see is that if you visualize cell death, you get the same pattern that you saw in the growth inhibition growth curve assays. So the Citrobacter cells are much more sensitive to hydrogen peroxide, but much more resistant to streptomycin when they have the PT. And the Salmonella, in either case, don't seem to care whether it's a oxidative stress or antibiotic stress. So these are really interesting. What we've done is a couple of things. The first time anyone has systematically profiled the effects of PT, as opposed to looking at a single organism, a single source of D&D genes. It's the first time someone has looked at sort of a panel. It's the first report of PT being involved in antibiotic resistance. And it's also the first report of the opposite effect of what the literature showed, where PT can actually sensitize to hydrogen peroxide. So all these are not only very interesting from sort of an academic standpoint, but there are also a number of important clinical implications for these. One of the most interesting being that it's widespread in clinical isolates, particularly Mycobacterium abscessus is a common skin and wound infection, and about half of all clinical isolates of it have PT. So there's the possibility that this should be something that we're routinely testing for, especially because it's relatively easy to confirm its presence or its absence with the electrophoresis. We also have some evidence from our own lab that PT is present in gut microbes. So essentially, if you do an LCMS analysis that we developed on feces, on DNA extracted from mouse feces, you get tons of signals for PTs. And not just the sort of 1 or 2 dinucleotide contacts that are well defined, but you get lots of dinucleotides, indicating that you have lots of different species in the gut that harbor PT. And that could be potentially important for things like restructuring of the microbiome after infection or after knocking down with antibiotics. This could guide the way that the microbiome sort of rebuilds, and that could have pleiotropic effects on human health. And so there are a number of questions still to be asked and answered, but it is very clear that phosphorothioate is playing an important role in the survival and the replication of these organisms. So, all total, we took a little bit of a garden path through the stages of infectious disease, looked at pathogenesis, diagnosis, therapy, in each case highlighting some ways that antibiotic resistance, diagnosis, inhibiting pathogenesis can all be targets for these modulated modified nucleic acids. And we have evidence these modifications of both DNA and RNA are playing important roles at each process and each stage in this, which is really nice. So it's clear they're critical, and it's clear that they are playing pleiotropic effects that we're only now beginning to understand in infectious disease. Quite importantly, this could not have been done without the selfless and relentless help of an army of people. This is but a small sliver of them here. So everyone in the DM lab has been fantastic. I've tried to pull out people that in particular helped me with specific projects and with specific things I talked about today, including a number of Europe students that were able to slug through some data. Tons of people in the Division of Comparative Medicine were absolutely indispensable for the animal work and in teaching this chemist how to do a little tiny bit of biology. And you guys for coming. That really means a lot to me. I'll close with some thoughts. This is a little less science. That feels hard. It's quite hard. Obligatory Wikipedia reference. These are the smartest and most impressive women I've ever met in my life. It's my mom and my grandma. Neither of them could be here. I like to think that if they could see this, they would say a couple of things. They would say it's about damn time you graduated. They would say, you worked with rats. And I hope that they would say that they're proud of me. They would say it was a long time coming, and it was a lot of really nice conversations, and they were happy that I was here, and in the very bad times, when I wanted to leave, they were having none of that nonsense. Anything impressive that I've ever done pales in comparison to what they did for me. Those dark nights when grad school was hard and when all I wanted to do was run away and be an ambulance driver for the rest of my life, this woman kept me in it. Alex, you mean the world to me. You have been my partner for many a year and many a year to come, and this could not have been done without you. And finally, since Pete brandishing a butcher knife, he unexpectedly took me in 8 years ago and couldn't get rid of me. Funny story, actually. So when I did the REU program in 2006, I was torn between applying to be in Pete's lab and in Steve's lab, actually. And so one of the things that I ended up doing that was a little silly, but I looked at profiles on the website, and you guys each had nice little headshots, right? And in headshot, Pete has a relatively round face, and Steve has a relatively tall face. And I thought, well, Steve is probably going to be the 6'5" gargantuan man that I'm going to be terrified of. Pete looks, a little bit more my shape. I'll go with Pete's lab. The first day of the program, Pete walks in, all 6'5" of him, and then Steve walks in. And I said, well, I think I picked wrong. Thankfully, things panned out. It worked out well. And I've gotten to work with both of them over the years, which has been really nice. Pete, you believed in me when I did not believe in myself. And that means a lot to me. So, to all of you, I can only say thank you. And bring it.
Well done. Questions? Yes.
How much does the rat study cost? In terms of like rats, like it just like you say it's low cost, and I realize it's low cost in comparison to especially VSL.
Yes.
But how much does that cost?
Sure. Yeah. So it's not low cost in sort of the abstract. It's a very fair thing. And because most of the husbandry work, the actual manipulation was done by us, that saves some work and that saves some costs. But between all total, the study, we actually did two. So we did an 8-week one for establishing the infection parameters, and then a different 8-week one for doing the biomarker work. And so all totaled, for the husbandry for all those weeks, the necropsy supplies, buying the animals, you're talking several tens of thousands of dollars. Not dirt cheap by any means. But substantially less than a cohort of rhesus macaques or BL3 housed rats or something like that, which would get probably more like a 6-digit number.
Okay, I'm going to bore in with a more detailed.
All right, bring it.
going back to the rat studies, the urine studies, the principal component analysis of all the RNA modifications.
Yes.
Did you only, and nothing stood out, that was the end result of that. Did you also include the unknowns? Were you only really analyzing the known guys where you had synthetic standards, or did you do everything?
Yeah, so the important point, I only looked at things where I actually knew what it was. I wanted to avoid pulling in spurious things where I was looking at tail fur or what I used to clean the cage with, or so all of the uncharacterized species, either things with question marks by them or just numbers with unassigned structures. I didn't look at those in the PCA. So there's the possibility that some of those unknown things.
It's right
yeah,
there
exactly. So there's absolutely the possibility that something we don't know, a structure, an unknown structure, is correlating with infection. So yes. So none of the, I will say,
start working on that tomorrow,
yes,
right?
I will say none of the unknown things appear only in control or infected, so they're not that good, but there is the possibility that some of the unknown things correlate.
The urine's still in the freezer. Right?
It is. There is still tons of urine. Tons.
Because that's a relatively low sensitivity approach.
Yes,
you took.
very much so.
You got the really loud, the abundant modifications. And if you were actually to go through and do very targeted analysis, you can get 3 orders of magnitude better sensitivity.
Yes.
And potentially pick up the bacterial specific or rare modifications that may have a better story to tell.
Yes. Yes. So we've done a little bit of targeted work. I haven't pushed the sensitivity, so I've done targeted analysis for the BCG nucleosides. None of those show up. But I haven't done UPLC yet, and I haven't really pushed it using the 1290. So I've done standard chromatography and MRM, and you don't see them. But I haven't pushed it sort of to the limits of sensitivity. I'm still, even in the targeted, I'm still in the several double-digit femtomole range, 20, 30 femtomole limited detection. So if you're talking about a bacterial nucleoside that's, I did this math actually, so if you had a nucleoside that was present on one tRNA and you had a normally dividing population of cells, do the math, you fudge a little bit, you end up with about 30 attomolar for a single nucleoside in the urine. So a little bit below the order of magnitude, but conceivable as physics improves.
So we need septimole sensitivities to get that kind of
Exactly.
as well.
Pete.
I just had a question on your urinary biomarkers. 2 questions actually sort of related. Like you were talking about profiles, now this include quantitation, and so what was your internal standard? And 2, I noticed that the volumes were very different. How did you correct for that?
Yeah, so in both cases, so to normalize signal intensity, all of them had the same final concentration of coframycin added, and so coframycin was used as sort of an internal standard for nucleoside-like organisms, or nucleoside-like structures ionization. And then to control for volume and differences in urine concentration, we did the Jaffe method of creatinine concentration, and I created a scaling factor for each one based on creatinine concentration.
And that's a lousy way to normalize for excretion. It's not there are some more sophisticated methods that are much more challenging.
Yeah, so there are some good mass spec methods where you can look at something like total signal normalization. We played around with that a little bit. The software we had to do it only worked with the QTOF, and we were using the triple, and technical limitations happened. We tried creatinine. We tried just sort of a dumb method of just OD 600, like for how yellow it is essentially, and that correlates decently well with creatinine. So we ended up going with creatinine.
That was my question.
Okay, cool. Eric?
Oh, so with the original H. pylori stuff, did you, so the genes that HP has, but you didn't detect the molecules.
Right.
So do we have standards for those, and could you develop like a
like a dilution method and
yeah.
Let's see here. So one of them is lysidine. We know it should have lysidine. I do not believe we have a standard for that. Other one that it should have is MNM5UM, which I believe was in the standards we got from the European lab. So that one we do have a standard for. And I think lysidine, Singapore has a standard for it, but we don't have here. So for at least,
do we have that one?
don't think so.
No. All right. So for at least one of them that we expect to see, we have a standard.
Right.
So.
Because it would just be interesting to know whether, because it's possible that it just shows up at certain times,
Mm-hmm.
and so then it could actually be even more interesting.
Yeah, that's true.
So I was just wondering.
Yeah, the fact that also if you go back to the chromatogram and look, so no uridine modifications show up and they fly the worst, and so my guess is it's simply a sensitivity issue, that I'm not picking them up just because I don't have enough. And things like lycidine are only on one tRNA, so it's going to be at a low level anyway.
All right, let's thank Brandon for a very well done seminar. So we're now going to proceed to the closed door defense.